Hi, folks. Test number 1, Essential 2 in Biology 12. It says, name each phase of mitosis and describe the most important events that occur in each phase. And so what I have here is I have a photograph of a onion root tip that we looked at in class. And so just kind of get some things out of the way. This cell here, in fact, this whole group here, these cells are all in interphase. And so they're not going to be included in our mitosis because remember, interphase is not part of mitosis. So what we're going to do is we're going to look sort of at a prophase. And what we would be looking for is a cell where we'd start to see chromosomes forming in the nucleus, but are not in any kind of order. So we might look grainy or tighter chromosomes. And when I'm looking around here, I see this one, I see this one. Even up here, this one, you can sort of see it, maybe a later part of prophase. So one of the things that you can't see here is that the centrosomes are now on both sides of this, and spindle fibers are forming. But what you can see is the nuclear membranes breaking down, and you can see that those chromosomes here are beginning to tighten up. So if I were going to put down, what's the most important things of prophase, I'd say, okay, well, prophase, and I'd be saying that the nuclear membrane breaks down. I would put that the chromosomes are supercoiling. And then I would put that the spindle fibers are forming and attaching to the chromosomes at the centromere, which is that center part of the chromosomes. So if I were saying, oh gee, what's most important for that phase, I think that's probably what I would put right there. Okay, and again, I'm looking at something like this guy here. I'm looking at probably this guy here. In fact, all of these guys right here would sort of all be sitting in some form of prophase right there. All right, next up, if you recall, is metaphase. And with metaphase, I'm looking for that nice line in the middle where the spindle fibers have drawn our cells toward the middle of the cell, that area that we call the equatorial plane. And there's a nice line right here, look at that. Where these chromosomes have all been sort of gathered toward the middle. I suspect this is also part of a metaphase, maybe a little further along, a little bit of anaphase happening there, but certainly this one we can see that nice line where those chromosomes have been drawn to the middle. And so simply put, in metaphase, there's not a whole lot that happens other than that. Metaphase, and in this case, bold that and underline. In that case, I would suggest that we are looking at chromosomes lining up. Chromosomes drawn to the center of the cell at the equatorial plane, which is the fancy biology word for the middle. Okay. And that would be our metaphase. Now, our anaphase, there's a lovely, let me circle the metaphase here before I get too far along here. There's our metaphase right there. My drawing skills with my mouse pad are not that great, I apologize. And maybe that other one right there, I'll use the mouse. Maybe a little better at that. Cool. Okay, now our anaphase is where our chromatids are starting to separate. And hard to find a better one than that one right there. You can see that the chromosomes on this side are being drawn to one pole, the chromosomes on this side are being drawn to another pole. You can see the trail edge here of these chromatids separated from one another. They've clearly vacated the middle, and in this case they're being pulled to opposite ends. So again, if I'm looking for some words here, anaphase, and this time I'll see if I can get that a little slicker, Control B, Control U, and there. Turn that off. Double-stranded chromosomes. Double-stranded. I didn't turn the double on. Okay. Double-stranded chromosomes are separated and drawn two opposite poles. Something along those lines. That seems to be not wanting to come off. There we go. And I checked my spelling. Opposite poles. Sure. And so that's a very simple way to put it. Now let's look for a telophase here. Telophase. Oh, there's one right there. And so I'll just highlight him. Here, right there, we've got a cell that we can see that there's a new nuclear membrane forming around here. The chromosomes haven't even quite unwound yet. You can still sort of see them. And this nice little line here is evidence that cytokinesis has begun. There's a nice end plate forming in between those two. And so I'll just kind of scroll this down a little bit. And let's type that in there. So telophase. Telophase. Yeah, that. Okay. Let's see if I can turn it off right this time. Yep. Telophase. New nuclear membranes are forming around the two new nuclei. And I'm just going to say that evidence of cytokinesis, that is that separating into two new cells, may have begun. In this case, it has. Now, be careful, because remember, this is an onion root tip, so we've got a plant cell here, and that's, you can see the evidence there of that new cell plate. Now, if this were an animal cell, then you would sort of see this. I'm going to see, maybe I can get a different color here. Can I? Yeah, right there. Let's say that I would be looking at animal cell. I would sort of see this kind of furrowing in like that, and it was sort of the hourglass that on the same side. I can't. That's not happening in a plant cell because the cell walls are cellulose. They don't blend in like that. Instead, what we have, and I'll erase that so you can see it a little better, is we've got that nice little cell plate forming across the middle that you can see there. And so that, clear evidence that cytokinesis has begun. Okay, I hope that helps you get those essentials, and talk to you later.